Good day everyone. Welcome to the course on biological data analysis and visualization with R. In the last few classes, we talked about basic statistical techniques. We performed some statistical tests hands on in R. Then we talked about correlation analysis and we followed that up with discussion on analysis of variance ANOVA. For each of these topics, we also took some real data sets and we analyzed them hands on in R. So, in this class what we will do is we will now talk about basic data visualization techniques. Although in one of the earlier classes I showed you how to actually generate these XY plots etc. Those are the very basic ones. We will now move on to slightly more advanced slowly, right, step by step into more advanced data visualization techniques, and we will start with simple ones today. So, that is the topic for today's class. We will talk about basic data visualization in R. Specifically, we will be talking about three types of plots. The first one is histogram, and I believe I have showed you some histograms in maybe one or two of the earlier classes, but that was I did not go into all the details, all the parameters that you can control and how we can customize these different features in that plot and what each feature means, right. So, that is something we will do today more systematically. We will take data sets and then we will analyze and generate these histograms, and I will also show you if we can compare two histograms and put them together side by side. So, that would be very helpful in case you are trying to compare two different samples. We will then follow that up with something called density plot. So, it is kind of similar to histograms, but the representation is different. We have a density curve that we actually plot, and again this density plot is actually very helpful if you want to compare multiple parameters, values of multiple parameters or multiple samples in one plot. So, we will talk about this density plot, and again I will show you how to customize different options etc. We will then talk about mean SD plot. So, this is one of the very commonly used plots, right. We generate them all the time, and so where we have the mean value and the standard deviation. So, for any biological assay, right, when you do some experiment, whether it is some lab based measurements of let us say growth or maybe gene expression pattern measurements through quantitative PCR, or even if you are working with RNA sequencing data, you would like to, for example, show these mean expression values of genes as well as the standard deviation, right. So, this is something that is very commonly used. Of course, there are other ones as well, for example, bar plots etc. Of course, we will not be able to go into those for the constraint of time. Okay, so, let us start with first with the histogram. So, what we do in histogram very briefly is that we look into the distribution of observations over beans, right. So, we have a parameter value and we divide that into beans and then we count, right, how many observations are there per bean, and that is what we get as the frequency. So, the y axis is frequency and the x axis is the value or the beans of a parameter that we are interested in. So, we can then similarly do this density plot. So, it is more like a distribution of values of a variable over a continuous range, right. It is not binning anymore. So, unlike histogram, there is no binning, it is a continuous range of values for a variable, and we look into the distribution of values of a variable over that range. Okay. So, they are ideologically they are very similar, but their representation is different. So, the functions that we use for histogram is hist. So, this is something you have seen already in one of those earlier classes, and so this is something we will use today to actually generate these histograms, but of course, there is a lot more there to customize, which we will talk about when we go into the real example. And for density plots, we usually use this function plot density, and of course, within these brackets we have a lot of parameter values that we can put. Okay. So, we will see this step by step how this works, and we will generate these plots. We will then talk about this mean SD plot, right, where we plot the mean and standard deviation. Okay. So, with this background, let us now go into R and let us generate these plots hands on. based on the data set that we have already described, right, the gene expression data set along with the gene expression variation, or what we call sometimes as expression noise. So, with that data set we will generate these different types of plots. For mean and standard deviation plot, we will use another set of data just for just to make it more interesting, right. So, let us go into R now, and let us start with the analysis of the results. Okay. Okay, so let us go to the console. Okay, so here is the R console, and we have the codes in here. Okay, so we will start with this data set that I have already showed you, this filtered merge data east. Okay. So, what we will do is we will generate some of these plots first, okay, and we will do the So, first we will do the density plot, and then we will do the we will do the histogram first, and then we will do the density plot. Okay. So, let us start with the histogram. So, the data we have to load first. So, let us see, okay. So, first look into the directory where we are. Okay, we are in data analysis in R. So, let us go into now the correct directory. So, our directory is class 9. So, here we are, and here, okay. So, what we will do is we load the data first, which is, let us say, okay, this is the data we have seen already. Okay, and we have the header. So, we have seen this data already, and what we will do is we will take any two, let us say, two sets of values, and we will try to generate this histogram. Okay, let us start from a very basic level. Okay, so, which is the histogram. This is the command, as I said. Within these brackets, we will have to give the values that we want to generate this histogram for. Okay. So, let us say we want to compare the distribution of this expression heterogeneity in these two medium, and let us see how close these distributions are. Okay. So, for doing that, we will use this data. So, this heterogeneity or noise data in two conditions, they are in these parameters, right, data $DMYPD. So, maybe we can check the names of these variables, and okay. So, as we discussed before, right, the average expression, these are the mean expression values in these two different conditions, YPD and ST, and these DMYPD and DMST, these are like the expression noise values in these two different conditions. So, what the goal of this analysis or the plotting is to compare visually, right, how this distribution of these noise values look like in these two different conditions. Okay. So, we will start with this histogram, and this is the histogram that you want to generate. So, this is the very basic command, right, as you can see here. Okay. So, we have generated this, and this is a very basic command, but as I said, we can make it much nicer. We can customize this, and we can observe two histograms in one plot, right, so that we can visually compare how they look like. Now, you can see this x axis are the values of this DMYPD parameter, this noise parameter, and y axis is the frequency, right. So, these are the beans that have been created, and then the frequency for each bean is now plotted, right. So, you can see this here clearly, right. So, you can probably see the beans, etc., here. Okay. So, what you can do is we can also change the beaning, right. We can make it we can make the beans much smaller, right. So, we do not want these wide beans, maybe we can create this smaller beans. So, we can change the beaning by saying the number of breaks that we want, right. So, maybe let us say we want 20 breaks. Okay. So, it does not change anything. let us increase this 50, right. So, you see this now the plot has changed, the meaning has changed. Okay. The other thing you will notice, the y axis is frequency parameter, right. So, frequency is simply it is counting, right, how many observations are there, right. So, you have these are integer values, right, you see 0, 100, 200, etc. But if you want to compare, let us say, two different parameters, the total number of observations may not be same, okay, between these two parameters. So, in that case what you would have to do, it is better to compare the probability or the density, right. So, we can switch that on probability by saying probability equals to true. So, one thing you can see here now, in the y axis this has changed, right, from frequency it is now density. Okay. So, it actually allows us to compare two different parameters, right. We put them in the same kind of scale, right. For example, instead of saying, so one parameter might have 500 values, another parameter might have 300 values, and if you just compare those distribution, frequency distributions, of course they will look very different because the number of observations are different. But if the moment you convert to density, we are saying, for example, the percentage, right. So, let us say 10% are in this bin, 20% are this bin. So, we are kind of bringing them to comparable scale. Okay. All right. So, we can do that. Now, what we can do is we can change, now customize this a bit more, right. For example, what we see is these values, right, the like what we have discussed, right. So, we can change the font size, like we have discussed this when we are discussing basic xy plots. We can change the x levels, right. So, this is, as I said, x level is, so CX axis, this will change the font size of these numbers on the axis. So, the noise in, for example, let us say YPD, right. So, this is the x level that we want, and y level is density is fine for us, but we want to increase the label size. Okay. So, CX lab, as I said, above 1 means we are increasing the size, below 1 means we are decreasing the size. We can also change the main title, right. We can say histogram of noise in YPD, right, and we can change the size of that title. Okay. So, we can increase the size, and we have changed this. Okay. So, this looks much nicer. Now, as I said, we want to probably compare this with another distribution. So, maybe we would like to put this histogram in one color, right. So, that if we use colors, different colors, it will be easy to compare these two distributions, right, and also see the overlaps. Okay. So, let us try that and see if we can change the color of the histogram. So, is color So, how do you define this color? We introduced this RGB scale last time in one of the classes, right. So, color equals to RGB 100 0.3, and then we can also change the, okay, let us try this first, and you see this looks pretty nice now, but maybe we want to get rid of that border, right, the black border which we want. So, we can say border equals to false. Okay. So, there is no border anymore. Okay. Now, on top of this, we want to plot the histogram for the DMSD. Okay. So, this is something we want to do. Let us do that. So, it is the same command again, right, DMSD breaks equal to 50, probability equal to true, because we want density, that is more comparable. We have now defined the axis etc. already, x level, y level. So, we do not have to define them again, right. What we need is the color, right, color for that new histogram that will be added here. Okay. So, we are using, let us say, blue color. So, that is why in RGB scale the first number is for red, the second number is for green, the third number is for blue, and then this is the transparency, right, how transparent you want that color to be. Okay. We build in transparency because then we can visualize this overlapping distribution. If there is any overlap, that will be visible to us. Okay, this is the histogram. I don't think we are missing anything. So what we will do next is we just need to add this, okay, because if we just do the histogram command, if we run that, it will replace this one and it will generate a new histogram. So we have to give this add equals to true command, okay. Oh, okay, so this generates this blue, but then what we see, we have not got rid of the border, okay. So what we'll do is then we'll modify this border equals to false. In that case, the black border will be gone, and now we can see this distribution, okay, and you can clearly see where the overlap is, right. So the overlap region appears in violet color, right. One thing you probably have noticed is for the blue distribution, we are actually the y scale is not enough, okay. So which means we have to increase the range of that y scale, okay. So we can say, we can increase this y scale by saying y limit, see, it's now 0.2, let's say we increase this to 0.3, okay. Okay, so this is fine for the red. You can see we have increased the y limit, right, range of y. Now on top of it we can add the blue, okay. So it looks now, okay, we are kind of accommodating, but if we want, we can increase this further, right. We can make it 0.35, let's say, and then add the blue, right. So we can try this out in different ways, and you can see what is the most suitable range, okay. You can actually do this for more parameters, right. So not just 2, but then very quickly we will see like it will become very cumbersome, right. You will not be able to see any of the distribution. So up to 2 distribution is fine, but beyond that it's not really good for visualization. The other thing you will also notice that x scale mostly 30 up to 30, right. Most of the observations are mostly 30. There are probably very few after that. So we can also change the x scale by saying x limit, okay. Anyway, I think we will leave this at that. So once we have generated this, so maybe can we save this plot, right. So because we have generated this very nice histogram, we want to show this in a report or paper or analysis, whatever, in a presentation. So can we save this, okay. So yes, we can. So let's, in this class, let's save this in PDF. We usually prefer to save in PDF files because it actually maintains the resolution, right. So you can then actually generate lower resolution files. So we usually prefer PDF or EPS, okay. So you can also save in JPEG, TIFF, etc. We'll see them in later classes, right. How do you do this? So let's save in PDF file. So the command is dev.copy to PDF file, and then we have to give the file name, which is histogram noise.pdf. The other thing we use with this when we are saving in PDF is we just want to maintain resolution, okay. So if there are points in this plot, they might change if you do not use this command, use thingbands equals to false, okay. So we want to maintain the resolution of the points and everything that's there. So we just type this, okay. So it has written to a file now. What you have to do, you have to say def.off. So it will switch off this one, and it will also close the PDF file and save this in the PDF file, okay. So this is the command to save in PDF, and then you have to type def.off so that the file is closed, okay, and this will also close, okay. So what you are seeing in this output, this should be written to PDF, and the file name we can specify, and this is for just maintaining resolution, okay. Okay, so this is done. We can later on check that that file has been saved, okay, going into that data analysis directory, and we can see here, okay. So this is the file that has been saved, okay. All right, so we have now done the histogram. So one of the things as I said to you is, okay, histogram is good for if you want to compare up to 2 parameters, two sets of values, but beyond that it becomes a bit difficult, okay. So what we use in that case, when you have multiple parameters, you want to check or compare their values visually, or you want to represent their values, the distribution of values, we use the density plot, okay. So density plot is you use the command density to calculate density of that parameter, right. For example, here we can calculate this density of this DMYPD, this is the noise parameter. It will just calculate density, it will give you the median value, etc., mean, etc. So, for x, y, right. So, what we will do is we actually use this information to actually generate the plot. Okay. So, here the x is actually defining the range and y is the density. Okay. So, let's try that. So, the command is plot density and oh sorry, there is a spelling mistake, right. So, density. So, this is how it looks like, right. So, it's an histogram, but without those bars that we have seen in histogram, right. Okay. So, very similar idea. So, it is just a line plot, right, showing the line. So, that is actually good for comparing multiple variables. We can now further customize this to make it much nicer. So, what we will do is let us first do the standard things that we do, right. We x axis, right, cx dot axis is we increase the size of these numbers that are there along the axis. Then we have the label, right. So, x lab is expression noise in YPD, let us say, right, or maybe just expression noise, because we will compare YPD as well as the ST. Then Y lab is fine, we want to keep it as density anyway. So, we increase the size of these labels by Cx dot lab equals to 1.4, we are increasing the size. Main title we can give, right, density plot of noise in YPD and ST. So, just say density plot of noise, and we also increase the size of that main title by saying Cx dot main equals to 1.5. Okay. Okay, so we have increased this is kind of standard practice for all of the plots that we are doing, but this line is actually very thin, it is not really visible, right, and maybe you want to put this in different colors. Okay. So, there are ways to do this. Again, color is here, we can simply, because it is line, so we can simply use a solid color by saying color equals to red, or you can also use RGB scale without any issue. So, we have mentioned the color. What we want is we want to increase the thickness of that line, right, and to do that you have to use this command line width LWD equals to 2 or 3, right. So, let us do this for 2, and you see the color is coming and the thickness of the line has increased. We can increase it further saying LWD equals to 3, right. Again, you can customize and see which thickness looks the best for your plot. Okay. So, the next one is we have to add the other distribution, right, for DMSD in the other environmental condition. Okay. So, that is what we are going to do now. Is for doing that, what we have to do is add lines, the same thing, instead of saying plot we have to use lines. We have defined the axis anyway already, so we do not have to add the axis anymore. By saying lines, we are saying add this line on top of this. Okay. Do not create a new plot, but just add this line on top of this. What we need to do is we need to define the color, which is, let us say here we can define a solid color blue, and the other thing we need also LWD, right, the line width. Okay. So, it is saying the density function is saying giving some error. So, which means, and what is the error? Let us look at the error. So, it says x contains missing values. Okay. What does it mean is that probably there are some NA values in this data $dm underscore st. Okay. So, we have to get rid of them. So, we can check, right, whether there are any values or not. Yes. So, the command is actually is .na, right. So, Is dot NA means it will check for all elements in that column whether you have NA values. Okay. And you see this is logical, right, true false, right. True means there are NA values in this parameter. Okay. So, what we can do now, we can use that same command, but say there are ways to do this. I think we can say NA dot RM. So, let us see if it works. NA dot RM means remove NA. So, with some commands NA dot RM will work, in some cases NA dot omit will work. So, let us see which one works here. Okay, so here it does not work NA dot RM. So, it will work NA dot omit. Okay, so, NA dot omit works, and you see that it will get the blue plot, right, blue line. Again, as before, we notice that this blue line is going above this y axis scale, the range that we have. So, we need to increase that. Okay, and to increase that range, we will use the y limit. Okay, so we have increased the y limit, then the command is y limit equals to c from 0 to 0.35, right, and now on top of that we can add the blue line. Okay, so this is something we have generated, right. This is the density plot. You can play around with x axis scale, etc., but this is kind of the idea. As you can see, you can now add more data on top of this plot, right. If you have, let us say, another noise measurement in another different condition, right, you can actually put this distribution here as well, right. So, you can add multiple distributions in different colors, right, and you will be able to visualize any change in distribution, right. So, this is something very, very useful if you have a lot of data and under different conditions you want to see the value of the same parameter under different conditions. Okay. All right. So, we have created the density plot. We can again save it as before, but let us will not save this for a moment. I have shown you how to do this. So, I will just type def.off, okay, and it will close. Now, we have now looked into the histogram, how to generate histograms and how to make these overlapping histograms, and then we have talked about the density plots. The other thing we will talk about are the mean SD plots. Okay. So, this is something that we want to generate, and let us see where we have this mean SD plots. Okay. Okay. So, for mean SD plot, we will actually use a slightly different data set. So, here I am showing you the data set. Let us see, let us load this data set. So, it is actually in cloud. Okay. So, okay. So, we have gone to the directory where we have that data, and this data file now is a different one. So, sorry. Okay. So, this is a growth curve experiment that for which we have data for 24 hours. Okay. And what we have is, okay, sorry, we have to give the header again. Without header, okay. So, here we have this. Okay. So, header file, it says this is time in hours, then we have different samples. Okay. So, what you see are the samples. So, I can check the sample names by saying names, right. Okay. So, these are the column names we have. The first column is time. So, because we are doing a growth curve experiment, so this is over time. Then you have G01, 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 right, 1, 2, 3. So, these are actually replicates. Okay. So, what we want to do is we want to calculate the mean and the standard deviation from these replicates, and we want to generate the plot. Okay. And similarly, we have done this experiment for multiple different strains, right, G02, G02, and again 3 replicates, G03, 3 replicates, and G1001, 2, and 3. For each of them, we have 3 replicates. So, what we want to do is we want to create this plot, right, where we will be able to see the mean growth of each of these strains as well as the standard deviation in our measurements. Okay. So, that is what we will do in the next class. So, we have introduced the data now, and we will see how we can use R for generating very nice mean SD plot. One last thing we want to show is how you can check this in a data frame in a very nice table. You can actually say this command view, just note this V is capital, only then this data will be shown in a table, right. You can see this, right, this the first column is time, then you have the columns that I just mentioned. So, you have 3 replicates for one strain, then the next 3 replicates for the other strain, and so on. And what is good is that you can scroll around and see the full data, right. If you just type this data here in the console, right, you cannot see the, it is very difficult to see the full data, the full table, right. So, this is how we can see the full data, and once it is done we can close. Okay, so, with that we will conclude. So, we have looked into the basic plotting in R, right, and we have talked about histogram and density plot. So, we have seen how we can customize these plots. Followed by that, I have just introduced the data set for mean SD plot, which we will be working on in the next class. Thank you.